Good afternoon and welcome to the USDA Human Nutrition Research Center on Aging's weekly seminar series. My name is Paul Jakes. I'm a senior scientist at the HNRCA and the host of today's seminar. And I'm very pleased to welcome and to introduce today's speaker, Dr. Zarin Machanda. Dr. Machanda is the Usen Family Career Development Assistant Professor of Anthropology and Biology at Tufts University. She received her PhD from Harvard in 2009 and continued postdoctoral research there in the Department of Human Evolutionary Biology until 2017, when she came to Tufts University. Dr. Machanda is currently co-director of the Kambali Chimpanzee Project, an almost 40-year study of behavior, physiology, and ecology of wild chimpanzees living in Kambali National Park in Uganda. Her main research questions center around the form, function, and development of social relationships. She also conducts research on communication and cooperation. Her current research focuses on the understanding of the social determinants of healthy aging in wild chimpanzees. Dr. Machanda is also on the board of directors of the Kasisi Project, an NGO that works to improve the lives of school children who live near the Kambali National Park. Today, Dr. Machanda will discuss some of her research findings from the Kambali Chimpanzee Project and presentation titled Patterns of Aging in Wild Chimpanzees. I'm certain there'll be a lot of questions today, and I would like to remind you to ask your questions using the Q&A icon located on the webinar toolbar, not the chat box. And I encourage you to add your questions as they arise during the presentation. And with that, let me turn the microphone over to Dr. Machanda.
Thank you so much, Paul, for the lovely introduction, and I'm thrilled to be here today with all of you to tell you about the work that we've been doing on patterns of aging. And I'm going to focus on both social and physiological aging, but most of the talk will focus on kind of the social aging patterns that we've been finding. So with this group, I don't actually need to talk too much about aging or describe kind of aging, but as all of you know, humans are living longer lives than we ever had in our evolutionary history. But a lot of the research on aging has really focused on the pathology of aging, right? Thinking about diseases of aging, things like Alzheimer's. And what we want to kind of think about is, humans are evolutionarily selected to have these long lives. And instead of thinking just about what are the diseases or the kind of pathologies that affect us when we're aging, we want to think a little bit more about what does healthy aging look like and what is kind of the evolutionary model for healthy aging. And to do that, I'm going to propose over the course of this talk and through my research that wild chimpanzees, who are our closest living relative, are a potential model for understanding what healthy aging might look like in a long-lived, large-bodied ape. And if we really want to think about using an animal model for healthy aging for humans, I would argue that chimpanzee life history data, or thinking about chimpanzees, are kind of incredibly essential data for us to have. So this is the classic kind of life history plot that we have that biological anthropologists use to document the different life stages across different primates. And for a lot of kind of life history stories about human evolution, we've almost always focused on these kind of early parts of life history. Things about, like the fact that humans all have this really long kind of period of infancy and juvenility compared to our primate cousins. But of course, the apes also, compared to monkeys and lemurs, have also really long periods of development. And that's usually related to the fact that it takes a long time to grow a large brain. What has often set humans apart is also this end of life kind of pattern of our life history, where we have this post-reproductive state after 50 that can last 2, 3, even 4 decades, where we have ceased our reproduction. And I'm not going to get into the evolutionary kind of arguments about this, but one of the most commonly used arguments is that the selection for the importance of grandmothers has really promoted this long post-reproductive life. When we look at non-human primates, we often kind of think of adulthood as this period after adolescence when these individuals are reproductively active. But if you look at the lifespan of a chimpanzee, they become an adult at 15 and then can live into the wild even into their 60s. And there's kind of an assumption that maybe all of adulthood, if you are a wild primate, looks the same. But really, chimpanzees are navigating, almost 3 or 4 decades of life as an adult. And what we see after about the age of 35 is that these chimps enter what we call kind of a past prime adulthood phase. So this is kind of when you start to see their bodies, they're losing muscle tone, their fur is just not quite as shiny, they're starting to lose teeth, they're going bald, lots of things that we might recognize as humans as kind of this period of old age. And what we see with chimps is that after about 35, they enter this past prime life, but then they might have to navigate this age category for maybe 2 decades, maybe something like 25 years until they die. And so we want to kind of think about what is happening to chimps in this past prime or their old age period, and how do they compare to the patterns that we're seeing in humans? And I would make a further argument that the data from chimps is kind of really interesting because of the number of shared traits that we have. So they live these long lives. Most people aren't really aware that they live till almost 60 in the wild, but they do, sometimes in captivity as well. They have those large brains and they live extremely kind of in these really interesting, complex social groups where they do form long-term social bonds with one another. And sometimes those social bonds can be positive and sometimes they can be negative. And I'll tell you more about that through the talk. I think one of the things that we can do with chimpanzee data that we might not be able to do with human data is that the kind of detail that we can get from chimpanzees far exceeds the kind of detailed behavioral and physiological data that we can get from humans. So as you'll see through the talk, when we go out and follow chimps, we go to where they are, where they went to sleep. They wake up, we get urine and fecal samples first thing in the morning from every chimp, and then we follow them for their entire day and we record what they're doing every single minute of the day, who they're doing it with. We collect every urine sample they have through that day, as much feces as we can carry, and then we follow them until they go to sleep at about 7, so we get about a 12-hour day, and then we get up and we do it all over again the next day. And so we wouldn't, I mean, it would be so awkward to do that with a human, right? To follow them around and record everything they're doing and then collect every urine sample. And so what we can do with chimp data is get this incredibly detailed understanding minute by minute as they age every day, almost of their entire lives. I like to joke, the kind of data that we can get is like, I know, for example, the average copulation length of every single male chimpanzee that we've studied. This is Big Brown, and his average copulation length was like 7.9 seconds across thousands of copulations. And that's the kind of data you should not have on humans, right? No one should know that about their friends or relatives. Like, you should not know that sort of stuff, but we do have that kind of power to get that kind of detailed data. I think the other thing about chimps that is interesting that we don't have to grapple with in the same way as with human studies is that we don't have to deal with cohort effects. So, for example, if you have a study with humans where you're comparing older humans with younger humans, there's nothing we can do about the fact that all of these younger humans have grown up with the internet and social media, or at least most of them, compared to all of the older humans who didn't have that as part of their social development. And so when we're trying to say something comparing the social bonds of older and younger humans, we do have one of these issues of cohort effects. So we can kind of think about chimps as being devoid of those effects. So as Paul said, the data that I'm going to show you comes from a long-term field site that I helped direct called the Kibale Chimpanzee Project. And since 1987, we've been studying the Kanyawara community of chimpanzees who live in Kibale National Park, Uganda. This was a site that was started by my PhD advisor, Dr. Richard Wrangham, in 1987. And pretty much and then today it's run by me, Melissa Emery Thompson, and Martin Muller, who are two professors at the University of New Mexico. And so all of this is a really big project where everyone is kind of involved in all of these projects as well. And so since 1987, for the most part, every single day since then, we've been getting up, collecting all of that long-term data, and it gets stored in my lab here at Tufts and then in a big relational database that I maintain, and that includes all of the behavioral as well as physiological and ecological records. So just to show you what it's like to follow chimps in the wild, I have some videos for you. This is a day where we were actually able to climb into one of their big fruit trees. This is a ficus or a fig tree. That's one of their big ones in their habitat. And so there will at any given time be, in a tree this big, maybe 10 or 20 chimps, and you'll see quite a few of them feeding on fig fruits. This is one of their commonly eaten fruits. And not only will you see them feeding here, but you'll also hear the sounds of the forest and you'll see some social interaction. The alpha male is somewhere over here, and he moves and spooks one of the lower ranking males. This is the alpha. Chimps are not quiet, so I think oftentimes the videos get muted by Zoom for being too loud. But you can see chimps all over. We can identify all of them from their faces as well as just the shape or features of their body. Here is a female who has a sexual swelling, which I'll tell you about in a few slides. Chimps spend about 50% of their time in the trees. We don't climb into the trees very often. It's just when we can. So most of the time, if they're in the tree, we're on the ground looking up, collecting data. But they also spend 50% of their time on the ground, and we can get pretty good detailed interactions, data on their interactions. So this again is the alpha male, and as he walks, he's going to actually pass by a subordinate male named 2K, and 2K is going to give this emphatic vocalization called a pant bark, which is kind of like like that, and that's the signal that I am subordinate, please don't beat me up. So I'll let you see it for yourself. Oh, the sound might not work. Zoom might have muted me, but this is a way that we can calculate all of the dominance hierarchies. So we can get the direction of these pant grunts, and we get thousands of these every year, and we can calculate a dominance hierarchy. So just so we're all on the same page about chimpanzees, chimpanzees they live in a multi-male, multi-female group. So we have lots of adult males, lots of adult females, about at any given time, maybe 12 adult males, maybe 18 to 20 adult females, and then the dependent offspring. We have, I think, 63 chimps in our community right now, but we never see them all at once. And instead they exhibit something we call fission fusion, which I'll explain on the next slide. Chimpanzees are frugivorous, so their main food is ripe fruit. Males are philopatric, meaning that they stay in the community that they're born into, whereas females will transfer at adolescence to a neighboring community. And the important part about this is that the males then have kind of strong, right, they're brothers and sons and uncles, and so they tend to have closer relationships with one another, they cooperate more, and they're more social. So a good chunk of this talk is actually going to focus on male social bonds because females stay relatively solitary. Females will kind of join larger parties when they have this sexual swelling, which is the swelling of the labial tissue, which responds to estrogen. So around 10 days of ovulation, around ovulation, they'll get this really big, pink, wobbly signal telling all of the males that they are ready to mate. And like I said, it's about 10 days. Lots of the males compete over these females who have swellings. So fission fusion refers to the fact that we have this multi-male, multi-female community, but we don't see them all together. And they do have a territory or a community range that they defend, sometimes to the death. And what we do have instead is that these chimps are broken up into temporary subgroups called parties. Now, parties with males are often bigger, whereas parties of females can just be maybe the female by herself with just her dependent offspring. But these are temporary, and so throughout the day, they will break up and come back together, or fission and fuse and reform. And the important part about this is that it means that chimpanzees have a tremendous amount of power over who they spend time with. So if one of the chimps is bothering someone else, so just say Frank's being really irritating, you can leave the party and maybe not see Frank for several hours, days, weeks, whatever you choose. So when we talk about social bonds, we're talking about, are you spending time in parties together? Are you sitting near each other in these parties? But just remember that because of fission fusion, chimps have a lot of control over who they spend time with. So as I said, we collect a lot of data at our field site. We collect these full day focal follows, which is following one individual in the community for the entire 12-hour day, recording what they're doing every single minute. And we try to get a full day focal follow of every chimp every single month. And that includes babies as well as adults. We have a second field assistant who is going along with the field assistant collecting the full day focal follows, but this field assistant is recording all occurrence behavior of everyone else in the party. So that includes all copulations, all aggression, all play behavior, anything unusual that's happening, like maybe if they're hunting, whatever we see. And then since 1996, we've been collecting health and detailed health data, going through every single chimp we see, if we see any signs of illness, disease, injury, etc. And then we have physiological records from urine and feces also going back to 1996. And then this data is all in a relational database. Just to show you, we're really old-fashioned. We collect data pen on paper, and I know people they're like, why do you do this? This is so time-consuming. But actually, we get chased by elephants about twice a week. Well, maybe not twice a week, but at least once a week. And if you're being chased by an elephant and you drop a tablet and it gets destroyed, that's way more expensive than dropping a clipboard and that getting destroyed. So we still collect data pen on paper. And then this is all, we have data entry staff who read through all these notes and extract it into spreadsheets for us. But just so you can see what this looks like, this is a day in May of 2024. All of the chimps have 2-letter codes. So AJ is Johnny, YB is Yogi Bear. So Johnny charged, chased, and then made attack 2, so a prolonged attack of about 30 seconds on Yogi with kicks and slaps, and Yogi flees screaming. Yogi has a dry cough. 5 minutes later, there's a copulation where the female screamed and ran away afterwards between Lonjo and Rwanda. That lasted 8 seconds, which is 1 second longer than Lonjo's average copulation length. In that copulation, the female approached. We have intergroup aggression, where we have distant calls and the party responds with wah barks. And in total, we have over 125,000 hours of behavioral data and physiological data on about 160 plus individuals. I say plus because we just actually had a baby born this week, so it's kind of constantly changing. And so we don't have a big sample size in terms of individuals. Like, it would be hard for any wild primate site to have thousands and thousands of individuals in their data set. But what we do have is almost complete full life histories and data across the entire lifespan of several dozen individuals that we've had over the years. In terms of physiology, we get most of our physiological data from collecting urine. And this is our field assistant, John, demonstrating our very, very high-tech urine catcher, urine technology, urine whatever. And it's a stick with a plastic bag tied to the end of it. So it's very sophisticated. The longer the stick, the less likely you're going to get splashback from urine. But because the chimps climb into trees, they'll pee usually after they've before they're about to leave. We collect the urine on these clean plastic bags. And from that urine, we're able to assay testosterone, estrogen, progesterone. So we're able to do all the reproductive hormones. Humans and chimps are so closely related, we can actually use human pregnancy tests in the female urine. So we can measure human chorionic gonadotropin. I will show you some data on cortisol later in this talk, but we've done a lot of work on cortisol and stress responses in the chimps. We can measure insulin and thyroid as measures of energy balance. We can measure creatinine as a measure of muscle mass. And we've done some oxidative stress work, and we have a assay on C-reactive protein that we're developing. We can also get salt from urine, but wild chimps, at least at my field site, don't have salt in their urine. So it's not really worth us measuring anymore. We also can get lots of data from feces, but I won't go into all of that detail right now, but please feel free to ask questions. So I'll start by showing you kind of the general idea of thinking about chimps as a model for healthy human aging. And one of the things that we really want to try to understand or unpack from our data is thinking about whether the human aging phenotype that we see is a result of the fact that humans have this greatly extended lifespan and potentially these unique lifestyle features, especially in, the United States, or, or whether or not what we're seeing in humans is actually a common feature of being a long-lived ape, a long-lived, large-brained ape. And so we're looking for similarities and differences between the chimpanzee and the human aging phenotype to see whether or not we can tease apart what is really responsible for this human aging phenotype. So I'm going to start by showing you some data on cortisol and how cortisol changes with age. So just so I should have probably had a slide before this, but humans, as when we look at cortisol patterns, human cortisol increases with age, and we also see a blunted diurnal rhythm of cortisol in humans as they age. So what I mean by blunted diurnal rhythm is that cortisol at the start of the day is very high, and then you get this kind of characteristic decline in cortisol through the day. A blunted diurnal rhythm means that you don't kind of see that characteristic decline quite as much. So what I'm showing you here is data from our chimpanzees by time of day. And what we've done here is, first, I'm just showing you the fact that males tend to have higher cortisol than females. That's partly due to the fact that males probably have somewhat stressful lives with all of the aggression and vying for dominance that they're doing. And what we've done in plots B and C here is that we've divided our adults into those who are under 35, who are in their prime, versus the older 35, over 35, or the past prime individuals. And what you can see from these two plots is that actually you do see that the older individuals do have elevated cortisol, and they also show that blunted diurnal rhythm. And I should point out that I didn't put it down, I didn't put it here, but this is not because older chimpanzees are actually receiving or dealing with more stress. We actually, in this paper, talk about the fact that they don't actually show that, they're not experiencing more stress as they age. That does not necessarily explain this. And so what we're suggesting here is that the human phenotype that we see, which also shows elevated cortisol and this blunted diurnal rhythm, is not necessarily this side effect of an extended lifespan or a byproduct of recent shifts in human lifestyle, but this is something that we're seeing shared with another large-bodied, large-brained ape, and might be just part of a normal aging process for us as a species. And so I think it's also probably likely that why we're seeing this elevated cortisol and blunted diurnal rhythm is partly because just the feedback on turning off cortisol after a stress response just becomes harder. So after a stress response, older individuals are likely just not turning off that cortisol as quickly as younger individuals. And that seems to be true for humans as well as chimpanzees. So another way that we've looked at kind of something that's more about physical aging as opposed to social aging is that we've also looked at frailty in wild chimpanzees. And frailty is defined as this loss of functional homeostasis. And I probably don't need to explain this to this group. You're probably all familiar with this. But the way we think about it is that it is the kind of individuals become worse at withstanding illness without loss of function. And so one of the ways that we've looked at frailty is to take the creatinine data that we have from urine, which is a reliable measure of muscle mass, to see if that kind of loss of muscle mass is predicting worse outcomes or changes in activity that would result in worse outcomes. And so one of the things that you can hopefully see for males and female chimpanzees is that they do lose muscle mass with age, right? So they aren't able to maintain, males kind of keep their muscle mass in these glorious days when they're fighting for dominance. But as soon as they hit that kind of past prime age of about 35, we start to see a pretty significant decline. Females are declining kind of consistently through adulthood. One of the things that we have seen is that when we look at whether physical condition changes, other things, so we kind of looked at physical condition and whether that has negative effects on health or survival or activity, we actually found that there was no effect of age on traveling or resting. There was an effect of age on time spent in trees and time spent eating ripe fruit. But the important thing here is that actually seems to be true of aging in general. Individuals who showed more of a deterioration in muscle mass did not have an increased change in activity. So we weren't kind of seeing that characteristic like loss of muscle mass is what's leading to that loss of activity. What we also found only for males is that extreme loss of muscle mass, kind of a sudden drop of muscle mass, predicted death in that same year. So if you had a sudden loss of muscle mass, you were likely going to die within 6 months to a year. But we did not find that for females. And so what we're seeing here is that there are some aspects of frailty in these chimpanzees where a sudden loss of muscle mass does predict this kind of acute mortality. But what we do not see is that loss of muscle mass is actually predicting any sort of long-term kind of loss of function or long-term, future mortality as we do in humans. So what we think is maybe more unique about humans is our ability to stay frail for a very long time, and that frailty kind of leading to loss of function over long term. We're not really seeing that in wild chimps. What we see instead is more this like acute loss of muscle mass certainly has an impact on their survival. Now, I'm going to move away from physical and physiological aging and actually focus more on the body of work that we've been doing that is about social aging. And I love this result, but this is from a paper by Holt Lundstad in 2010. I know it's getting to be old, but it's one of my favorite aging papers. It was a meta-analysis across over 300,000 people that looked at what predicted kind of survival or what predicted kind of mortality. And so you can see that if you quit smoking versus continue smoking, quitting smoking increases your, or decreases your mortality. This is not rocket science, not drinking versus excessive drinking. Also, abstaining from alcohol has this protective effect on your mortality. But what was kind of surprising about this paper is the impact or the effect size that social bonds and social relationships had on protecting human mortality. So that individuals who are socially integrated, for example, or individuals who have very strong social relationships had a 50% increased likelihood of survival than people who had decreased social relationships. So obviously that means that there's something really interesting about social bonds that are protecting us from mortality. And I always tell my students that this clearly means that you can absolutely smoke and drink, but you just have to make sure you're doing it with friends, right? So that's clearly the lesson that this paper teaches us, but don't actually listen to me about that. So social bonds matter for humans. But there's also some really interesting data out of psychology that talks about how social bonds are changing or how people kind of change as they age. And one of the things that we see is that older individuals have what we call a positivity bias. So they're more likely to interpret situations in a positive way. They're more likely to just view their social relationships in a positive way. We also know that when people are young, they're really focused on building new relationships, potentially to get information about how to build relationships, who's going to be a good friend, who's going to be a bad friend. And so young adults have these big, big social networks, but in fact, often that comes with maybe some negative social interactions or negative social relationships, partly because we're learning how to navigate our social world. As you get older, you don't need to learn anything more about how you make friends or who's going to be a good friend or a bad friend. And so as we get older, we shift, our social networks shrink because we get rid of those bad friendships and we focus on emotionally satisfying relationships. Now, this is true cross-culturally, these kind of patterns of social bonds mattering and this positivity bias and this shift towards more emotionally fulfilling or, stronger and more positive bonds. When you read the psychology literature, the reason that psychologists would say that humans show this pattern is that it's because we can monitor time and that we understand and have an awareness of our mortality. And so the idea here is that because we're humans and we understand that there is an end to things, as we get older and death is kind of the ultimate end to things, we start thinking, I really don't have time, literally don't have time to keep these bad relationships in my life. I'm going to get rid of them. I'm going to focus on things that make me happy. Now, some of the best evidence for this is the fact that patients who are given a terminal diagnosis in their 20s or 30s actually shift their social bonds to look more like old people. And this is also true of any time horizon. It doesn't have to be death, but I always think about when you're a freshman in college, the way you make friends and prioritize your social bonds is very different from your last week of college, which we're in the last week of college for many of our seniors right now. Now, chimpanzees cannot monitor time and are very unlikely to have an awareness of their mortality. It's hard for us to know for sure, but we know that lots of cognitive tests show that they don't monitor time like we do. Their sense of time is kind of on the order of hours, maybe days, but nothing like what we see in humans. And so for some of this work, what we've been really thinking about is how can we think about chimps as helping us understand this particular explanation? Because if chimpanzees and humans show the same pattern of social aging, we then have to kind of reassess whether or not this is about these subjective time horizons or whether there could be other neurological or psychological mechanisms that would result in this pattern. If humans and chimps show different patterns, then this would actually provide comparative support for this idea that human social aging is about the ability to monitor time and be aware of our mortality. So we actually published this in Science. This was like the greatest life achievement of my career, where we got the cover of Science. But it's actually not an old chimp that they picked for the cover. We didn't get to choose the cover, but they picked this like 25-year-old chimp. He just has gray hair. Anyway, so this is work that I have done with my colleague, Dr. Alex Rosati, who's a psychologist at the University of Michigan. And so for this project, we used long-term data. We used 21 years of data. We had 21 male chimps. We focused on males because they have those stronger bonds compared to females. And we had quite a number of years for each individual in this study. So the first question is whether or not chimps show that positivity bias compared to, are they showing kind of a similar positivity bias? Now, one of the challenges of this kind of work is obviously how do you translate human data where we can ask people questions and ask them to come into a lab and interpret social situations? How do we translate that into something that we can do in the wild where we're just collecting observational data? But chimpanzees, they're pretty complicated and they do exhibit what we would kind of consider positive behaviors, which are affiliative behaviors like grooming each other, as well as negative behaviors where they are fighting each other, right? So what we did is actually looked at the kind of relative rates of those positive behaviors and negative behaviors. So in blue, we have grooming. This is social grooming, where two individuals are sitting closely together, not fighting each other, and just engaging in a affiliative interaction. What you can see is there's very little change in grooming with age. It's slightly positive, but not significant. But they're kind of pretty consistent with grooming as they age from 15 to over 55. I think the oldest chimp in our sample here was about 56. But what you do see is a really strong decline in aggressive behaviors. And aggression can be non-directed, which are actually just dominance displays where they're running around screaming, throwing things, trying to intimidate everyone, or directed aggression where they actually have a target and they kind of physically aggress against that target. And so what we're showing here is that their grooming kind of stays relatively the same, slightly increases, their aggression decreases. So as they get older, they're reducing their negative behaviors and keeping their positive behaviors fairly similar. And we're saying that basically what that shows is this shift towards more kind of relatively more positive behaviors. So making the argument that this is evidence of that positivity bias. What about actual social relationships? So one of the things that we can do with our data is actually kind of look for what we might call the equivalent of friendships. And these are individuals that you spend most time in close proximity with. This is, if we followed humans around, this would be a pretty good metric to figure out who your friends are. And we can do this in two different ways. We can kind of classify friendships as either lopsided, which are the ones where I'm always kind of seeking you out as a partner, but I'm not your number one partner. So it's kind of, I often think of this as like the younger sibling dynamic, where younger siblings are often chasing after their older siblings, but it's not reciprocated. So we can think of mutual friends as these kind of reciprocal relationships where you're both equally interested in each other. And what we found is a really strong age effect here, where everyone has about 4 friends, right? Almost every male chimp will have about 4 friends, but the quality of those friendships really shifts with age, such that individuals who are over 40, 35, 40, are much more likely to have or maintain those mutual friendships as opposed to those lopsided friendships. And so the ratio of those friendships changes quite a bit with age. And that, again, is showing this shift towards those more high quality mutual relationships. What's interesting when we did this data is that everyone seemed to really want to be friends with the old chimps. And I'll get back to this a little bit, but this is the proportion of mutual friends. So many of the older adults were mutual friends with other older adults, but even the prime-aged adults really seem to seek out these older adults as friends. When we look at lopsided friends, the younger adults are kind of desperate to be these friends with the older guys, but they're less likely to have those bonds reciprocated. So just the idea here is that these old individuals seem to be quite attractive, and they seem to be quite attractive to each other. So when we're out watching some of our old guys, this is Stout and this is Big Brown. He's our 56-year-old. He is about 52 in this video. He actually died when he was 58. And he didn't die of old age. He actually died because he got gored by a bush pig. So he went out in this like blaze of glory. But it's not uncommon to see our two old male chimps just sitting next to each other, quietly grooming each other and investing in this kind of mutual relationship. So what we found in that study is that there was this shift towards this positive affiliation, positivity bias, and selectivity. And that this is actually happening in wild chimpanzees without that sense of time or mortality. And so we make a couple of arguments in the paper for why. I'm not suggesting that this isn't happening in humans, that we're not thinking about our mortality, but there might be alternative mechanisms to get to the same kind of social aging phenotype. For those of you wondering, we do not find the same results in females. We did a slightly different study where we looked at how female social aging occurs using grooming networks. And actually, females become much more solitary and less social with age. They kind of mostly focus on their offspring as they get older and kind of disconnect from the larger social group. So we kind of make an argument that this might be because social bonds are just not as important for females, and that we might need to consider in primate aging studies sex as a really important variable that affects how social aging is occurring. We also did a review paper where we kind of compared chimpanzee data with what is found in other primates. Most of that data comes from old world primates like macaques and baboons. There's very little that we have of other apes like gorillas, which is unfortunate. And some of the new world primates like capuchins, who also lead relatively complicated lives. But what we found is actually that chimps are quite different from other primates, especially those monkeys. Whereas monkeys actually seem to have an increasing negativity bias. They decrease their affiliated behavior with age and increase their aggression. That's different than what we found with chimps. There's also kind of limited evidence of selectivity in the way that we are showing it with the chimps. So not necessarily that kind of focus, that real kind of like focus on these mutual partners. And then we are suggesting that we're also finding kind of similar patterns of sociality favored in the philopatric sex. For monkeys, we see kind of increased sociality among females, whereas with the chimps, we're finding the opposite. And that's kind of interesting because most human studies don't find as many strong sex differences in aging with some of the same kind of social metrics that we're using here. I just want to quickly to kind of just leave you with some follow-up data and stuff that we're working on now. I wanted to just go back to that idea that male chimps, as they get older, are kind of socially attractive. And one of the things that we're actually is interesting is that that's not because they're more dominant, right? So male dominance rank peaks at about 25 to 30 when they're in their prime age. And then it decreases with age. So when we're talking about those old males being attractive, it's not because they're the highest ranking individuals in the group. I've already shown you this, but we already know that muscle mass peaks at 30 and declines. So they're not the strongest individuals in the group. And in fact, you can kind of, as they age, you can see this is Yogi. You can just kind of see that he's not as muscly and beautiful, and like kind of, you can start to see their shoulder blades through their skin. They're clearly not at their peak physicality. We know that their testosterone is decreasing, so we can get testosterone. We probably have about 20,000 urine samples in this plot where we can show that testosterone for each individual kind of peaks at about 20 and then has a decline. This goes hand in hand with the fact that they're losing muscle mass. But, and this is important, they do not decrease their copulation rates. So whether they're with Paris females, who are like the sexy sought after females, the ones that have already had babies and are older, or the nulliparous females, who are those first time, they haven't had a baby yet, they're just swollen for the first time, they're not particularly attractive. Males maintain their copulation rates with both kind of females, even the really attractive ones, well into their old age. And in fact, in our data set, we know that about 25% of the offspring are sired by old males because we can do paternity testing from fecal data. So that means that they're decreasing in rank, they're losing muscle mass, they're not as aggressive, and yet somehow they are still getting opportunities to maintain their reproductive success well into their old age. And what we found is that this probably is because these friendships are incredibly important for these males to obtain that reproductive success. What I'm showing you here is that they actually they maintain kind of pretty high coalition rates as they get older. But remember that some of their aggression is decreasing, right? Their display aggression is decreasing with age. Their directed aggression is decreasing with age. What we're showing here is the proportion of aggression that is done in coalitions. So as they get older, they are prioritizing doing coalitions, doing coalitionary aggression. And what I mean by this is that two old males come together, or an old male and someone else come together, and together they can fend off a single higher ranking male and have access to a swollen female who was being guarded by that male. And so likely the friendships help you form these coalitions. Through coalitionary aggression, you are still able to maintain your high reproductive success. And we think that's kind of particularly important for males to maintain these mutual friendships. I'll just finish off with some new data that's not quite, it hopefully will be published this year, so keep an eye out open for it. But I just wanted to highlight that when the data I presented before is just time spent in close proximity to one another. So we were just kind of looking at this one measure of affiliation. But actually, not all friendships are the same. You can be friends with someone, but also have lots of fights with them. And we would maybe consider those to be frenemies, right? You're always grooming, but you're always fighting. Whereas you might just be friends where you're grooming a lot, but you actually have a pretty, you have a pretty good understanding that you're going to sit next to each other and maybe not end up fighting because your rates of aggression are quite low. And we think this is kind of an important distinction to make with the chimp data, because we know this is also true for humans, that not all marriages are good, right? So just being in a marriage does not actually protect your health and longevity. And in fact, people who are in healthy marriages do have positive impacts on their health and longevity. But actually, if you're in an unhealthy marriage, you actually have worse outcomes, worse health outcomes than if you just stayed single. And so what we did in our data is actually classified those friends as either kind of high, they're all high grooming, but some of them have also high rates of aggression, so we'd call them frenemies. And some of them are friends, where you have high grooming and low aggression. And so what we find is that it is really these high grooming, low aggression friendships that increase with age. Males of all ranks, low, medium, high, show a shift towards those really high quality, prioritize those high quality bonds. And here's the kind of really interesting thing. One of the physiological impacts of this is that the number of high quality friendships you can have between 0 and 3 as you get older, predicts cortisol. So if you have 3 high quality friendships as you get older, you basically don't show that increase in cortisol that I showed you at, earlier. And it seems to be graded. So the more friendships you have, the lower your cortisol production. What's really interesting here is that as you have more of those frenemies, those low quality friends, you're still grooming a lot, but maybe you're also fighting each other a lot. The number of low quality friendships predicts higher cortisol. So if you have a lot of friends where you're like, I just don't know about that one, as you get older, that's really what's predicting your cortisol. And actually, you have higher cortisol if you spend time with your frenemies than if you spend time with enemies, who are just individuals that you fight a lot. So it really seems to maybe be about the uncertainty of the bond that you have with these frenemies that might be driving a stress response. And we're going to look into this more, but it might be something about that kind of stress response, or, that lack of stress response with friends that is kind of driving a protective effect on your health and mortality. So just to finish off, just to say that we are showing that chimpanzees share patterns of aging, lots of ways. The cortisol data that I showed, the frailty. Older males are showing a positivity bias, have more friendships, so also showing similar patterns of human social aging, and that maybe this is linked to cortisol. And I think that what ultimately we're able to say is that a lot of the human patterns of aging might actually just be the fact that we are a long-lived ape, and they are shared with our closest living relative, and not necessarily just because of a more recent change in our lifestyle, or even just selection on that really extended lifestyle. And I think that some of these changing social dynamics are actually healthy and allow these males to maintain their reproductive success into old age, and maybe a really natural and evolutionarily selected part of aging. What can we learn from this? Well, I think if we kind of think about some of what we're seeing with humans, that decrease in social network size, the focus on just a few small friends who they know, what we can do is think, well, maybe when we see human social networks shrinking and people saying they don't want to make new friends, they really mean it. My mom doesn't want to make new friends, and my efforts to have her make new friends and go to the mall are wasted because it's not what she has. It's not part of healthy human aging. And maybe what we should do is actually focus more on getting seniors closer to the friends that they have, giving them opportunities to reconnect with some of their friends that they already have. I'd also just like to highlight none of this data, you can't do this data without long-term research. And so the value of our data is that we've been collecting it for almost 40 years. And this is one of those examples where you can't necessarily just take a snapshot. We can't just take 1 year of chimp data and find out these patterns. We actually have to have that 30 or 40-year data set. And it's worth investing in this kind of long-term research. With that, I will just thank the co-directors, all of the many, many collaborators, but most especially the field team that collected this data. We have an amazing group of Ugandan field assistants that go out every single day. They're incredible. And then our many funders, including the NSF and NIH. And thank you for listening.
Well, thank you very much for that very interesting presentation. We do have a number of questions in the queue already, so let me start with the first question, and that comes from Joe Mason, forgive me, and he says, thanks for sharing these wonderful observations of yours. Regarding the generalizability of your observations to other bands of chimps and even to humans, to what extent do you think these observations are specific to this particular band of chimps and this particular ecosystem in which they reside? For instance, would the same behavioral and aging characteristics apply to a band of chimps living in the jungles of Central or West Africa?
Well, it's a great question. I mean, we don't know the answer. I can tell you that some of the other aging studies that have been done have mostly been done in East Africa and show roughly the same patterns that we're seeing. I think in West Africa, maybe Central Africa, but in West Africa, some of the social dynamics are different, where females tend to have stronger social bonds than the East African females. So you might expect some of that sex difference to maybe be different or reduced. But I'm not anticipating that much difference across different habitats. Chimps across different habitats show fairly strong similarities in their social organization, in the impact that male social bonds have on reproductive success. So I wouldn't expect that much. I think captivity could be different because, again, you have kind of a different social milieu where the chimps can't get away from each other in quite the same way. So they might end up having to navigate social bonds differently. We do have a study at a captive site in Uganda called Ingamba, where we've been collecting social behavior data for the last 5 years. So not long enough to maybe show the long-term patterns. But we do see some sex differences there where the females are more social. I would anticipate that maybe the social bonds might, you can't get away, so you might have to repair the bonds in a different way or more frequently. So I don't think it would be that different, but I think captivity might be a bigger difference.
Okay, the next question comes from Roger Fielding, and he says, very interesting work. And his question is regarding the sudden loss of muscle mass and risk of death. What is the cause of death in these animals? Do they get cancers, for example, which could accelerate the declines in muscle mass?
I have never heard of a wild chimp getting cancer, but to be fair, we don't always get. So let me step back. When a wild chimp dies, there's a good chance that we will not know the cause of mortality, because what happens is that they just, they just don't show up, right? We just don't see them for 6 months, and then we don't ever actually recover a body, especially because they're fission fusion. If we don't see a chimp for a day or 2 days or 6 weeks, we wouldn't necessarily know that they're dead or just going somewhere else. So a lot of the time we have no idea what kills them. When we do know what kills them, the kind of leading cause of mortality are human respiratory illnesses. And we do have often these outbreaks of respiratory illness. In 2013, I think we had 55 chimps in the community and we lost 6 in the course of a week to human rhinovirus. So that's something. When we have done necropsies or when we've managed to get a body of a chimp, we haven't found, cancers or things like that. I mean, they have a lot of adhesions of their organs to the kind of inside body cavity. They certainly have broken and healed bones and things like that, but nothing that any of the vets have said kind of resembles cancer or anything like that. So I think wild chimps are actually pretty healthy. They get killed by falling out of, they fall out of trees. Big Brown got gored by a bush pig. So illness, they also kill each other, right, in these intergroup interactions. So, but I don't think we would say that I don't think there's any evidence that they have like cancers that would lead to that sudden onset of muscle mass. Sometimes they might, I think what I've certainly seen is perhaps some kind of like tooth abscess that might make it very hard for them to eat. And then they just kind of all of a sudden lose their muscle mass. And it's that kind of acute loss of muscle mass that then predicts kind of a quick death coming soon over the, in an order of months, but not years.
Thank you. Sarah Booth asks, she says, thank you for a wonderful talk, and says, the data on frailty across the age range is fascinating. There is considerable variation and change in frailty, similar to the data from human cohorts. Have you explored the factors contributing to the variation in physical aging in chimps?
I wish we could probably do more, but I think this is where we get limited with sample size because I mean, we have over 160 chimps in our data, but we don't necessarily have data on, right, if they've been, not every chimp lives into their 50s. And so we just don't have enough of a sample size, I think, to explore that variation, which is why we have to keep going for another 50 years and keep getting that kind of data. So I think it would be really great to know, but I'm not sure that we can explore it in much more detail.
Next question is from Saidas, and she says, interesting talk, and asks, have you compared the rate of loss of functional homeostasis in these chimps in the wild with non-human primates from the Wisconsin and NIH monkeys, despite the behavioral differences, or with humans?
No, I don't think, I mean, I don't know. I have not, and we have not. I mean, we have been trying to do some work on metabolism, looking at, I mean, the way that we can measure metabolism in the wild is just by looking at respiratory rates. We can video them, record them while they're sleeping and measure respiratory rates. And we are showing a decline in metabolism, and then we've tried to compare that with captive animals, including gorillas, but we've never done anything with frailty with the captive monkeys in the US. I actually think maybe the more interesting data for us might be with the space chimps, the chimps that had been in the US Air Force as part of the space program, because there is a tremendous amount of physiological data taken from those chimps. But as far as I know, it's still all classified. So I don't have access to it. When I was a grad student, I spent several days trying to cozy up to the Air Force and get that data, but it is not available. So I think that would be interesting to see if, I think what you need when you're thinking about humans and apes, whether this is an ape phenomenon, just a human and chimp phenomenon. I mean, you need all of these outgroups. Like we need data on gorillas as another ape, another social ape as an outgroup. And we need data, kind of very similar data on monkeys, and maybe not just macaques and baboons, but like all of the monkeys, to see where is this on the primate lineage? Like, is this an ape phenomenon, or which might make sense because they're much longer lived than the other primates. Although not necessarily gorillas, where macaques are living into their 20s and maybe as old as 30, they're not living into their 60s like chimps. So I mean, the comparison is necessary, but hard to do. I mean, we're already wrangling with our own data. It's kind of hard to wrangle with other people's data as well.
Okay. The next question comes from Brown Monk, and Again, thank you very much for the interesting talk. Is the human ability to stay frail for a long period of time due to something specific in human biology or to medical interventions?
I think that's a great question. I think maybe there are people on this call who would know the answer to this better than I do, but I would make the argument that from an evolutionary perspective, one of the arguments about that prolonged frailty has to do with the fact that we take care of each other in our old age, right? That we offer social support, but also like if someone can't forage once they're too old, we actually feed them. And that's just not something we see with other animals. We don't share food and they don't share food in that way. They don't provision older individuals. They don't even wait for them. So I think from an evolutionary argument, the argument is that because we take care of each other, we can have these older populations. And so obviously another explanation would be that we have those medical interventions, but even independent of medical interventions, if we're thinking over evolutionary time, the fact that there are signs even in the archaeological record and the fossil record that humans were cared for after injury suggests that we are able to kind of deal with that frailty.
Okay, and the last question we have today is from Chris Wiley. And Chris says, thank you for such a stimulating talk and topic. In monogamous species, cortisol is often associated with blood glucose levels in response to anxiety. In chimps, do you see blood glucose differences in response to social hierarchy in males relative to females?
So yes, so there are sex differences as well as rank differences. So we tend not to compare. I'll kind of tell you what the data say, but when we look at something like cortisol, what we see is that generally high-ranking males have the highest cortisol. So there's kind of this stress of being dominant. For females, it seems to be more the opposite, that low-ranking females tend to have higher cortisol, but that's being driven mostly because they have kind of access to poor food. When those females have sexual swellings, the higher-ranking females tend to be more attractive, which means they get a lot of aggression directed at them, which means that their cortisol goes up. So it can be context dependent. In terms of C-peptide or kind of we can measure C-peptide, which is kind of a marker of insulin. We see high-ranking females tend to have the higher C-peptide, which is an indicator that they have better quality food. The high-ranking males also tend to have higher C-peptide. So I do think that rank and then the sex difference, we haven't really directly compared those two things because I think it's a little bit hard to know what it would mean. I mean, chimps are kind of stressed across the board. And I would say if you read our cortisol paper, we show kind of the different social contexts for males and females that induce a stress response. So for males, it's the presence of a swollen female, but also rank instability. So when there's some kind of rank turnover, these males get stressed. And for females, it tends to be kind of their own rank, but also how much aggression they're receiving from males tends to be kind of the thing that really drives their stress up. I don't know if that answered the question, but there are lots of sources of stress for chimps. They're going to be different than what we see for monogamous primates, where the biggest source of stress for monogamous primates seems to be separation, and that is not really the case for these chimps.
Okay, well, thank you very much. And as all the questioners said, is a very interesting presentation. Thank you very much.
Thanks, Paul. Thanks, everyone.